Hi, this
Hello, how
is
are
a
you?
Science Hangout from Science Mission. We have today Dr. Andrew Ewald, Associate Professor of Cell Biology from Johns Hopkins School of Medicine with us. Let's look at Dr. Ewald's achievements. I'm going to highlight only a few of them as it takes a long time to go through all his honors and achievements. In 2011, Dr. Ewald won American Association of Anatomists Morphological Sciences Award for outstanding contributions to the field of epithelial morphogenesis. In 2012, he won American Cancer Society Research Scholar Award. In 2014 to 2017, Dr. Award became Fellow Keith Porter Endowment for Cell Biology for exceptional contributions to cell biology. In 2015, he won Keynote Speaker Gordon Research Seminar Directed Cell Migration. In 2015, he won Society for Photo-Optical Instrumentation Engineers Systems Biology Pioneer Award for developmental epithelial organoids as a platform for tissue-level systems biology. In 2015, again, Dr. Ewald won Metastatic Breast Cancer Network Research Leadership Award for expression of our basic understanding of the biology of metastasis. In 2015, he won Jaron Green Foundation Scholar Award. In addition, Dr. Ewald won several other awards. Also, he has published articles in several highly reputed journals, and also he is in the editorial board of several journals and has contributed tremendously to various conferences throughout his career. Welcome, Dr. Ewald.
Thank you. It's great to be here.
So can you please talk about your background, research interest, your education, etc.?
So I grew up in a suburb of Philadelphia with two brothers, Carl and Jonathan. And I went to college at Haverford College, which is a liberal arts college just outside of Philadelphia. I studied physics there, actually, and gradually moved towards biology. I moved towards molecular biophysics while I was a student there, but stayed with my grounding in physics. I did my doctoral work at Caltech in Scott Fraser's laboratory in biochemistry and molecular biophysics before moving to UCSF to study epithelial biology with Zena Werb. I founded my own laboratory at Johns Hopkins in 2008 and have been here ever since.
Okay, so when did you decide on moving to cancer biology?
That was when I was a graduate student. I was working on the molecular control of cell migration, both of individual cells and of groups of cells. And I was working on this in the very early embryo, in the amphibian embryo. And I came to realize about 2/3 of the way through that many of the same questions I was interested in, because biologists always stay focused on questions. I could ask these same questions in mammalian epithelial organs and in mammalian cancers. And I sort of got the bug. It was kind of interesting to say, could I do the same thing in a context with more immediate relevance to human disease?
Like, I know you are very busy with your work, mentoring people and all those things. If you get some time, what do you do? Like, what are your hobbies and other activities you do normally?
I spend a lot of time with my family. I have two children, Michael and Eleanor, and my wife Shannon. So that's my first hobby, if you will, is my family. This summer we spent a lot of time at my daughter's dive meets. She's becoming quite the diver. In terms of my own hobbies, I really like bike riding, so both road and mountain. And I'm fortunate to live in an area of Maryland where you can really do both very readily. Just roll out of your house, and you can get to either one very, very quickly.
And like so far you had a pretty bright career. And can you please name some of the people who supported you, mentored you, coached you during this?
I think the two most important ones were my graduate advisor, Scott Fraser, who really helped launch my career and really believed in me from day one when I was a physics student who really didn't have a lot of insight into or knowledge of biological systems. So he really, and we continue to talk to this day. He's really been a tremendous supporter of my career. And then Zena Werb, who again saw me as a embryologist with a heavy physics bent, and was able to see that that could be a very useful skill set and set of approaches to bring to studying postnatal mammalian development and disease. I think in both cases they have a real gift for seeing what's not just what is today, but what's possible in 5 or 10 years. And I've really tried to learn from that. Here at Johns Hopkins, Denise Montel was the one, and Peter Devriotis were the two people who hired me here. And again, they had to look at where I was and not just assess where I was, but where I could be in 5 years. And I think that that took some vision, because we've made tremendous progress the last 5 years, but we didn't have everything working or in place when I was first hired here. And I think that's a unique strength at Johns Hopkins, is the ability to have very, very smart people with very, very good judgment really look carefully at candidates and say which ones have the greatest potential for future work. And I was fortunate that they gave me that opportunity.
And what are the things motivate you every day to work and put your 100% in there?
So I would say I have two different driving forces, and it's reflected in the different projects that we push. So the first is just fundamental curiosity, right? I want to understand at the cellular and molecular level how do organs form, even if it had nothing to do with any human disease. Even if we couldn't have any practical implications from it, I would want to understand at the cellular level how you build an organ, how you build an animal, how these things change over the lifespan. Fortunately for me, those questions do have deep relevance to human disease. And I would say we have another half of the laboratory which is fundamentally oriented towards understanding metastasis and understanding the specific disease processes that are driving patient mortality. So I would say we have a more fundamental half of the lab where we're really asking questions in developmental systems, and a more applied half of the lab where we're saying, what's really affecting the patients the most? What's the step we need to understand if we're really going to eventually change patient outcomes? So I have a dual motivation of fundamental curiosity and a very applied, almost engineering level problem solving bent of, what does the disease really mean? How do we figure out how to fix it, right?
Yeah. And can you please talk about your lab people, grad student, postdocs, and all?
Sure. I've been terrifically fortunate that some really wonderful and wonderfully intelligent people have wanted to work with me from the first day I joined here. I think the thing that people don't always appreciate until they hold a professorship in biological sciences is how dependent you are on your ability to recruit top people. And I've just been incredibly fortunate from day one. I've had fantastic graduate students. I've graduated 3. I've had 2 postdoctoral fellows leave the laboratory. They've all gone on to wonderful training positions for the students and professorships for the 2 fellows. I currently have 4 Ph.D. students in my laboratory, 1 postdoctoral fellow, a couple of undergraduates, and a research specialist. So we remain very graduate student heavy.
And can you please talk about some of the research projects going on in the lab?
Sure. Would now be a good time to turn to the slides?
Yeah, sure.
Okay, I would love to. So we're a microscopy lab, so it's really most fun when we can show everything that's going on. You'll give me just a moment. Okay, can you just confirm the slide looks good?
Yeah.
Fantastic. So we're going to talk about a series of publications that we've put together over the last 5 years, trying to understand how breast cancer invasion and metastasis works, as I highlighted or previewed, by iterating back and forth between normal development and direct study of disease. So our model system is the normal breast and breast cancer. And so we want to really start by saying the key challenge here, if we could understand one thing in breast cancer, it'd be metastasis. Because the surgeons will tell you, and the surgeons I work with here have told me, that they have primary breast cancer well understood. They understand how to get it out. They understand how to work with their partners in radiation oncology in order to treat the patient and really eliminate that primary tumor. And it's the spread of tumors to distant sites and the outgrowth of those cancer cells at distant sites, which is really what accounts for more than 90% of the mortality from breast cancer, and in fact, more than 90% of the mortality from human cancer in general. So that's easily said. We can say metastasis is the most important thing, but how does it work? what are the key steps and how could we possibly intervene? So at one level, it's easy, right? So breast cancer begins in the epithelial cells, the mammary ducts. Just to orient people, this is a mammary epithelial duct. The dark purple is the cells lining the duct, so the luminal epithelial cells. The pink is certain types of milk proteins, and the big white circles here are adipocytes or fat cells. So that's what a normal duct looks like in a normal mammary gland, in this case of a mouse. And if we turn on one gene, in this case the middle T oncogene, which drives deregulation of the PI3 kinase signaling pathway, bang, one gene equals formation of these huge tumors, poorly differentiated, highly proliferative. And in 100% of the mice with that transgene, they'll get invasive cancer. 95% will have lung metastases. And there was a very rich period for the last 30 years of testing different genes and different concepts and really trying to understand what is a sufficient signal, what is a single gene which can actually drive first cancer formation and then metastasis. The problem is simply knowing that parts list, knowing that genes A, B, C, and D are all capable of forming a tumor or capable of driving metastasis, doesn't actually tell us what's occurring at the cellular level, how it's being informed by molecular programs, and how it's being existing in dialogue with various mechanical and metabolic signals. So what I really strive to understand is how cells spread through the body during metastasis and how what are the different molecular control points that we could manipulate to disrupt the process. And I have to say, when I first transitioned from early embryology to studying cancer biology, I was really surprised how many seemingly fundamental and basic questions in metastasis really weren't understood to the level of resolution of early embryology. We knew lots of things, for example, about amphibian embryos that we still don't understand about metastasis in mice, let alone in people. So what are the barriers? It's not that there's a lack of interest. The first fundamental problem is metastasis happens deep inside the body and is inherently a whole body phenomenon. It's really accompanied by changes in systemic health, changes in systemic molecular signals, and involves transit between different organ systems. In animal models, metastasis occurs over months to a couple of years. In humans, it's years to decades. So it's a very long time scale. It's not convenient for the types of experiments we're used to doing in yeast and C. elegans, where the whole experiment start to end might be a day or two. And the third issue is that they're highly complex. Tumors are highly complex. It's not just a bag of cancer cells, but it's a highly complex alternate organ, and it differs from normal breast tissue in very many ways. And it's actually been just conceptually difficult to understand which of those differences is most important. So what have we really done? What's our angle on this? There's a lot of smart people working in cancer biology, and so you can't ever claim that you're doing everything better than other people, or even as well as other people. So what have we really tried to contribute? We've tried to develop in vitro assays to model discrete steps in metastasis in a way that makes them more studyable. And so the key thing here is we can develop these complex three-dimensional assays to make a key step in metastasis like tumor growth, invasion, dissemination, or intravasation observable and manipulable. We're able to compress the time scale, things that would occur over months to decades, we can model in days to a couple weeks. And because we have that, then everything in a three-dimensional culture environment, we can vary individual parameters like the composition of the stroma, how many fibroblasts or macrophages, the mechanical signals, the extracellular matrix, in very defined and systematic ways to understand their individual and combinatorial importance. So I would say when we started on this in 2003, the guiding lens through which people viewed metastasis was in terms of invented cell behaviors, that a breast tumor, pictured on the right here with the cancer cells in green, was discovering or inventing through oncogene-driven programs the ability to migrate and leave the tissue. Whereas the normal quiescent epithelium was really fundamentally locked down, non-motile, non-proliferative, not even really apoptotic, simply sort of frozen. And that metaphor of a static normal epithelium and a dynamic cancer epithelium has really persisted for several years. And I have to say, I found that dichotomy unsatisfying. My first entree point into this was actually to study the normal process of branching morphogenesis. So if we look here, we're looking at 2 mammary glands from mice. So at 3 weeks of age, you see the stromal tissue is close to adult size. And then there's just this rudimentary network of epithelial ducts. There's this primary duct that goes out to the nipple, and then just a handful of ducts going out, only partially filling the fat pad. You come back 7 weeks later, and there's this huge expansion in size and complexity of the ductal tree. And I really wanted to understand this at cellular and molecular detail. So how does that, how can you do that? You can study as many of these images as you'd like from the in vivo situation, and that can be very informative. But if you want to understand how the cells move, how the cells actually interact to elongate the duct, it's not enough. We needed an experimental model system so that we could directly manipulate and observe. So the classic approach here would be two-dimensional cell culture or tissue culture, in which you would harvest the organ, isolate the cell type of interest, and then put it in a petri dish. So we can do that. And here's an example of a piece of epithelium that's been settled onto a glass cover slip. And you see it spread out, the cells migrate, they divide. It's kind of an interesting movie, actually. What they don't do is rebuild that three-dimensional architecture of a ductal network. So you can look at this movie as many times as you like, and you're not going to see tubes forming, elongating, bifurcating, which isn't at some level surprising. We've taken away most of their normal signals, and we get a different answer. What I think was surprising, what really catalyzed my shift into this world of postnatal mammalian development and into this world of 3D culture, was how little you need to add back in order to get a really remarkably accurate, not perfect, but remarkably good model of the in vivo process. So we're going to do the same thing here. We're going to harvest the organ, in this case the mammary gland, chop it up and digest it with enzymes, and we'll generate these pieces of epithelium that we put into 3D culture. I want to highlight that the 3D culture models have been developed for most organs in many disease states. So this is a general phenomenon or a general paradigm shift in the field across many different organs, many different disease states outside of cancer, that people are trying to build these three-dimensional models. And the reason is, if we look at one of those same pieces of epithelium in a three-dimensional gel, it's truly striking the extent to which we're able to model the in vivo process of branching morphogenesis. You're seeing new ducts initiate, elongate, bifurcate, and polarize. And I want to highlight that per normal mouse, we can get 2,000 to 5,000 of these structures. Per mouse tumor, 10 to 50,000. And even from human clinical samples, we're getting 500 to 2,000. So it's this incredible expansion in our ability to ask and answer questions with tissue from the same genetic source. We're also able to couple this to really state-of-the-art imaging techniques and resolve how tissue change is accomplished by the coordinated and competing actions of different cellular populations. What we're looking at in the middle here is all the nuclei in this tissue with all of the plasma membranes in this tissue, and we're able to track in 4 dimensions with digital image analysis. We're able to track how those cells move. Then I want to call your attention to this image on the right, because I think we had a pretty static view of normal epithelium, and just that first view of the branching process gives you an idea that it's really much more exciting and interesting than that. But this particular cell actually changed our research program, because when you look at it, what you see is that it's actively migratory, extending protrusions, retracting protrusions, and if I took away the red channel of that normal duct, it would be easy to say it was a fibroblast migrating through a collagen gel. So we have all these key features that I would have thought were specific to metastasis: loss of apical basal polarity, collective migration, extensive proliferation in a normal tissue. And the key difference here is that it's migrating within the epithelium, on top of other and between other cells, rather than in the stromal matrix. So what that single cell tells us is that this paradigm of breast tumors inventing migratory behavior really isn't the right lens through which to view the problem. Instead, what's interesting, what's really fundamental, is why do the normal epithelial cells, with all this molecular and cellular migratory machinery, why do they stop at the tissue edge while the metastatic cells are able to leave? And what's great about this question is that it's really well suited to genetics, right? So you have these normal cells, they acquire migratory behavior in response to receptor tyrosine kinase signaling. They go to the tissue edge and they stop. Okay, great, this is what genetics does well. Let's just start breaking things until we can get the cells out. And it's also something that enables us to understand the microenvironment, because we can change different features of the microenvironment until we can see which cells, what are the signals that allow cells to leave. But first, our starting point for this, what we wanted to do first, was understand just if there were cell intrinsic molecular differences between this migratory epithelial cell and this migratory epithelial cell that leaves. So in order to answer that question, we developed a new set of three-dimensional culture assays starting from primary tumor tissue from mice. So same idea, you harvest the tissue, in this case the tumor, chop and digest. We get 10 to 50,000 of these pieces. We can then put them in three-dimensional gels, in this case a protein mixture called matrigel, largely laminin-1, collagen-4, and intactin. And this is when we got our first big surprise, really conceptual surprise of this project, which was we're taking tissue, and I'll remind you, this mouse model, 100% get invasive cancer, 95% metastasize. We take an organoid out of that tumor, and you'll notice the edges are clear. There's actually aren't any single cells migrating out into the matrix. So it's very difficult to ask and answer this question of how do migratory normal cells that stay differ from migratory cancer cells that leave when both of them stay inside the tissue. So what we knew was that human breast tumors could disseminate and metastasize. And I went and I talked to a bunch of medical doctors at UCSF. I started this project at UCSF, and I talked to some medical oncologists, and they said, well, we've got a real simple solution for you here, which is that you're studying mouse cancer, and our patients have human cancer. So if you would just study human cancer, right, then we would, these problems would go away. And so we took that as a challenge, and we did. So we developed a new set of techniques so we could start from fresh primary tumor tissue, chop and digest, get these same pieces now from the patient's primary tissue, primary tumor, directly into culture. We've done over 100 of these in the last 2 years. And we put one of those pieces. In this case, the patient had stage 3C breast cancer. 20 out of 20 lymph nodes are positive, and an organoid from that patient, when placed into 3D matrigel, it grows, but it's not disseminating into the matrix. And it's really hard to think about how metastasis works if the cells don't leave, right? And we had the absolute gold standard tissue. This was the right tissue. We've done this now more than 100 times with different patients. So we knew there was something. What that meant was that there was something missing. We had the right tissue, so it had to be missing from our assay. It also meant that we had something wrong with the conceptual model we were carrying around, that I was carrying around in my head about invasion and dissemination. I was thinking of cancer invasion as this big switch in a wall, and you flip it on, and you're going to get invading cancer cells in any microenvironment. And we clearly shown that wasn't true. There must be some feature in the microenvironment which was actually important to the cells being able to invade and disseminate. And the challenge here is that tumors in vivo exist in a really complex microenvironment. There's many different types of signals, and there's very many, thousands of changes as the cancers progress from starting in a normal duct to a frankly malignant tumor. And so it was challenging to think about which was the key difference here. Was it the fibroblasts, the macrophages, the TGF-beta, the mechanics, the collagen? What was the key thing? So we read a lot of papers. We really focused in on the single concept of collagen-1. Which is a structural protein, most abundant protein in the human body, and it's particularly enriched at the tumor stroma border. If you look at the image, the gold shiny signal on the upper right is fibrillar collagen-1. It's a Picrocereus red staining. And so we knew it was accumulating at the tumor stroma border. We knew that it was a very different molecular environment than the environment that a normal duct would face. And so we wanted to test whether this was important. I need to say that this microenvironment here is also rich in a large number of other signals. That's also where you would see cancer-associated fibroblasts. It's also where you see TGF-beta. It's also where you'd see a variety of different molecular signals, which highlights the strength, of the three-dimensional culture environment, which is that we can take individual patient tissue and then can just change one of those variables and test whether it's important. So what we're going to do here is take tissue from a single patient, generate a large number of these organoids, and randomly allocate them to two different microenvironments. The one we've been using up till now, which is this basement membrane protein mixture that's similar to the environment around a normal duct, or the stromal environment that would surround a breast tumor, so rich in 3D collagen-1. The great thing is it doesn't matter what you think about these two choices at the beginning. It only matters whether you see a difference. If you're taking tissue from the same patient, randomly allocating it to two microenvironments, if you see a difference between the two, then you know you have a key variable. So I hope the viewers can appreciate that we're seeing a large difference in invasive behavior between the piece of the tumor grown in 3D matrigel and the piece of the tumor grown in 3D collagen 1. And again, I need to repeat, collagen 1 is the major protein they're going to see at the tumor stroma border. And so we think this is actually a critical signal collaborating with molecular events in the cancer cell to drive invasive behavior. So we conclude from this section, in this collection of papers, that the extracellular matrix in the tumor microenvironment is a really critical regulator of invasive and disseminative behavior. But we don't think it's the only thing that's important here. In fact, we think it's very important to understand how oncogenes and tumor suppressors regulate cell behavior in tissues. I think there's a tremendous multiple decades of work identifying oncogenes and tumor suppressors, and I think we have a pretty good understanding of the major ones in the common cancers. What I don't think we understand sufficiently is how those oncogenes and tumor suppressors regulate cell behaviors in intact tissues. So we're working on that problem, that general problem of oncogene regulation of cell behavior in a variety of contexts in my laboratory. But the one I want to share with you today, the specific example today, is this idea of dissemination, which simply means escape. It means we do something to this blue cell, and then this blue cell leaves. We wanted to understand systematically what are the types of signals that could drive that. And for the next few slides, we're going to stick to that repressive matrigel environment and try to identify molecular signals strong enough to make the cells leave into the matrigel. So I did a lot of reading. That's how we start all our projects. Did a lot of reading, trying to figure out what other people had learned and what they were thinking. And dissemination was really being argued as working from one of two possible mechanisms. You either lose cell-cell adhesion as the trigger for dissemination, or you activate a broader program of epithelial to mesenchymal transition. These were typified by mutational loss or epigenetic silencing of E-cadherin, or expression of transcription factors like twist, slug, or snail. These were thought to be conceptually related because twist, slug, and snail were thought to repress E-cadherin at the transcriptional level. So I read all these papers, but what I wanted to really home in on, and I didn't think was out there, was this idea of whether loss of E-cadherin was sufficient for epithelial dissemination. And sufficient is really a specific term of art from genetics. It means that it has to be capable of driving epithelial dissemination in the absence of other changes. So we wanted to start from normal tissue, not tissue that had very many mutations already. So the basic logic here is we're going to take a floxed E-cadherin allele, so it's genetically engineered so that we can conditionally remove it. We'll isolate epithelium, and then we're going to use Cre recombinase to delete E-cadherin, either globally or mosaically, within the tissue. I'm going to show you the examples from the 3D culture. We also did parallel experiments in vivo. So what's the effect of deleting E-cadherin in all cells? We're going to run this in our simplest assay, where we start with epithelial organoids that are freshly isolated. And a couple days later, they're going to form an epithelial cyst with a nice lumen, nice polarity and junctions. And then the question is, with tissue from the same animal, when we delete E-cadherin, what happens? So what you see is the E-cadherin positive cyst has a nice lumen, nice apical junctional complexes marked by things like ZO1 and E-cadherin, right at that edge there. And when we delete E-cadherin, notice it's gone on the Western blot. It's very efficient global deletion. What we see is that lumen is gone, the junctions are grossly altered, but it's still an adherent group of cells. So loss of E-cadherin in a total form was not sufficient for dissemination in 3D culture or in vivo. But what about mosaic loss? Cancer isn't generally a change in the entire epithelium synchronously. So what happens if you lose it in some of the cells but not others? So we're going to use an adenovirally delivered Cre to get a mosaic deletion. And what we see in green here is the E-cadherin, is the Cre positive cells. E-cadherin's marked in blue, so you can see directly that they're E-cadherin negative. And you see that the E-cadherin negative cells sort to the apical surface of the tissue, that's right here, inside the lumen, and they sort to the basal surface of the tissue, but they're not disseminating. And again, we did parallel experiments in vivo with the same conclusion. So what we take away for this is that a model for cell dissemination driven solely by loss of E-cadherin is incomplete. It doesn't actually explain how a cell gets away. It explains local invasion, but not dissemination. Which raised a conceptual challenge for us on this question of the epithelial to mesenchymal transition. Because twist1 was thought to work. It does many things, but it was thought to regulate dissemination by downregulation of the epithelial phenotype, specifically loss of E-cadherin, loss of cytokeratin. So again, we like to start from very simple questions that are very precisely asked in an experimental system. So is expression of twist1 sufficient to induce dissemination? And we're going to do this using a tetracycline inducible model, so that addition of doxycycline will actually drive twist1 expression. So again, tissue from the same mouse will either leave twist off, then we have normal branching morphogenesis, or we'll turn twist on and figure out what happens. And there were three possibilities. It could do nothing. It could drive, it could exactly phenocopy E-cadherin loss, so you get local invasion, or it could drive dissemination of cells out of the epithelium. So again, tissue from the same mouse, same media imaged at the same time. The only difference is expression of twist1 on the right. So I hope you can appreciate that we see a striking and very acute dissemination of epithelial cells out of the tissue. And actually, if you watch it the second time, let's focus our eyes maybe right around here. If you watch the second time, what you're going to see is some of those structures, actually some of those cells go on to form new epithelial sites in the matrix. This isn't a tumor, and this isn't the complete process of metastasis that has to occur in vivo. But what this is showing is many of the cellular steps of the metastatic cascade under the control of a single transcription factor. And I may lack imagination, but this is the biggest change in epithelial cell behavior that I can imagine. You see a migratory cell with extensive actin-rich protrusions at the front, where only hours before it had been an epithelial cell within an epithelial group attached by intercellular junctions. So this is clearly a very potent signal. So next we had to do some control experiments. They're incredibly important to think about what are the right controls and then to do them and think hard about the answers that come out. So the first simple question was, is twist1 expressed in the cells even after they've migrated? So blue is twist1. We see nuclear twist1 in the cells that are out migrating through the matrix. And we also see nuclear twist1 in the cells that haven't left. So we're working hard on that problem too. Why do these cells express and localize twist1 without migrating out? But then the next question I had, which was very simple-minded in fact, I just wanted to know were these luminal epithelial cells or myoepithelial cells, the two major cell types of the duct. And so I asked my student to do the wrong experiment, which was to stain for cytokeratin 8 and 14. 8 for luminal, 14 for myoepithelial cells. And then she did this experiment, and she found that cytokeratin 8 positive cells disseminated and cytokeratin 14 positive cells disseminated. So we got a clear answer, but then I had to stop and think and remember that it shouldn't have worked, right? The EMT model, as proposed, argues for loss of cytokeratin, downregulation of cytokeratin expression. So these cells out in the matrix should have been cytokeratin negative, and they weren't, which caused us to think through a few more steps, like what is the status of their intercellular junctions, or their junctional proteins? So we did a whole series of experiments where my student really carefully captured cells just at the moment before they were going to disseminate, right after they disseminated, and after they were migrating through the matrix. And she was able to show that they were E-cadherin positive, alpha-catenin positive, beta-catenin positive, as well as cytokeratin positive. So these key hallmarks of the epithelial phenotype are being retained, which caused us to wonder what happened to the mesenchymal transition, right? This is clearly an epithelial cell that still thinks it's an epithelial cell, even though an EMT transcription factor is causing it to migrate. Which then drove us to the next rather heretical experiment, actually suggested by colleagues at the Cell Contact and Adhesion Gordon Conference, which was to test whether E-cadherin could possibly be functional in this system. Because they said, hey, look, E-cadherin is at that cell-cell border, and it's in a patch on the rear. Maybe it's actually in some way contributing to this dissemination process. And I say heretical because the EMT model is proposing that loss of E-cadherin is driving dissemination. And so further loss of E-cadherin by, for example, knockdown should accelerate dissemination. Conversely, it was possible that E-cadherin was required for dissemination, and then we would see less. So we identified hairpins that efficiently deplete E-cadherin. We allowed the protein to degrade, and then we turned on twist1. And what we got was a really big surprise. So on the left, with luciferase knockdown, you see that the cells are vigorously migrating out as single cells into the matrix. And if we knock down E-cadherin and then express twist1, we essentially eliminate single cell dissemination. If we look at one of those over time, what we see is strands, collective invasion strands, migrating out into the matrix, but that front cell, while highly migratory on its front half, is unable to release its connection to the rear. So these are not normal tissues in any way. But what we found, surprisingly, was that E-cadherin is required for dissemination. So an adhesion receptor is required for the dissemination process, which doesn't sound at all like the Elmer's glue sort of model of adhesion proteins that so many people carry around in their heads. So we're working hard to try to understand how that works at the molecular level. So we think this assay is going to really provide a platform to study the molecular basis of metastatic cell behaviors, the loss of polarity, invasion, migration, and then proliferation to form secondary sites. And our laboratory is working in 3 directions on this problem. On the molecular basis of increased motility, what are the signaling events downstream that actually drive this migration process? How does the disseminating cell release from its neighbors? And how does a luminal epithelial cell escape past that surrounding myoepithelial layer? So we hope to have answers to all 3 of those questions in the next 2 years. Now I want to shift gears in the final portion of the talk to really talking about frank tumors. So the previous section we were looking at cancer-inspired genetic events, but in the context of normal epithelium. And now we're going to engage with tumors in their full complexity. And I would say this project stems from my inability to look at a slide like this and understand which are the key cells for metastasis. We started off saying metastasis was the major driver of patient mortality. Now we look at a slide like this, which are the cells that are actually doing that work? Is it everyone? Is it the dark spindly cells, the pale round cells, the cells infiltrating the muscle, cells infiltrating the fat over here? Or is it the cells that are sitting near the immune infiltrates in the middle of the tumor? And that's just on morphology. If we come in with mutational analysis and transcriptome analysis, we look at different signaling events, we're going to find there's dozens to hundreds of phenotypes here. And this is really what people are talking about when they say tumor heterogeneity. And it's very difficult just by cataloging that heterogeneity to understand which are the key players from the point of view of growth, metastasis, and therapy resistance. So that's where we think it's really critical to develop functional assays that allow the cells to tell you which are the key events. So, simple question here. Took a lot of thought to get to it, but a simple question in the end. We know that breast tumors are highly heterogeneous. What we wanted to understand was whether all of the cancer cells were equally important for that dissemination and metastasis process. What we realized is we could prospectively identify the most invasive cells in a tumor by putting these organoids in that collagen-1 environment and simply asking, what's the molecular phenotype at the cells at the front? This is conceptually analogous to the Boston Marathon. If you walk to the front of if you walk to the starting line of the Boston Marathon in the elite runners division, everyone looks super fast. They're all wearing running shoes. They're all in really good shape. They're all going to complete the marathon in a good time. But which one's going to win? It can be very hard to predict. Instead, if you go to the finish line, if you go to mile 26.1 and you look, it's very easy to predict the winner of the Boston Marathon because he's in front, right? So same idea here. We're going to hypothesize that the cell leading the invasive process is better at invasion than the cell in the rear. For the next series of slides, it's important to know that there's 2 cell types in the mammary gland, basal and luminal, so a surrounding basal population, which is marked by keratin 5, 14, P-cadherin, and P63, and a luminal population that lines the duct, defines the duct really, marked by K8, 18, and E-cadherin. So the first mouse model we'll use here, the MMTV-PYMT mouse model, is a mouse model of luminal breast cancer. So it really isn't surprising that cancer cells express luminal epithelial markers. It's in fact a tautology, a circular argument. It's called a luminal model because of its widespread expression of luminal markers. What was surprising, so we checked very many times, was that the cells at the front, specifically the ones invading and reaching into the stroma, were actually expressing basal epithelial markers as well. Genes like K5, 14, P63, P-cadherin. So the first question we had was whether this was a feature of cells migrating in a collagen gel, or was there also a feature in the tumors from the same mouse line. So we figured out how to do these large-scale 3D reconstructions. And what you'll see here is blue now is marking the luminal cancer cells, red is marking the muscle, and green is marking the basal cancer cells. So the first thing is clear is that blue outvotes green. There's far more luminal cancer cells than basal. But when you go in and look at which cells are actually leading the invasive process, more than 90% were marked by these keratin 14 positive basal epithelial program. So the next question we had was whether this was true in additional mouse models or a feature only of a single model. So we got mouse models for HER2 positive and basal breast cancer to complement the one we'd already been looking at. And you see when they place them in culture, consistent with their behavior in vivo, 3 really different invasive programs: minimally, moderately, and highly invasive. Because they were models of different subtypes of breast cancer, it would have been very reasonable, I would say expected, if they were characterized by different molecular programs. We were quite surprised to find that, in fact, the same genes are expressed in the leading epithelial cells in each of these 3 models. We then went on to validate these in patient samples, both in fresh tissue and in retrospective studies. And we consistently find keratin 14 positive cells leading the invasive process across the molecular subtypes of breast cancer. So what we next wanted to understand was whether these keratin 14 positive invasive leader cells were a stable cell population or an inducible molecular phenotype. So is it a small number of cells that we need to target and eliminate, or is it a molecular program we need to antagonize? And the reason we were interested in this question is if we looked over time in culture, we saw a dramatic expansion in the number of K14 positive cells. So FACS analysis in vivo revealed that these were about 1 to 2% of the tumor. Consistent with that analysis, they're rare at day 0 in culture. However, you come back at day 4, and there's very many of these green keratin 14 positive cells. We could broadly imagine 2 possibilities. The first was widespread proliferation, and the second was phenotypic conversion of luminal phenotype cells to basal phenotype. We disentangled these 2 possibilities by using a drug called afitocolin, which blocks cell entry into mitosis. You see it works very effectively. We have essentially 0 mitotic nuclei. Despite that complete inhibition, we still see this large increase in the expression of keratin 14 in the epithelial organoids over time, suggesting that it could be phenotypic conversion. We then directly tested that concept using a keratin 14 promoter driving GFP we got from Elaine Fuchs's lab. What you can see here in real time is luminal phenotype cancer cells acquiring a basal phenotype. And it's these newly K14, newly green cells that actually lead the invasive process. So we have a molecular phenotype. The final thing we wanted to do in this particular study was test whether keratin 14 was required for the invasive process. Is it a risk marker, or is it actually a driver of the invasive biology? So we did this by taking late poorly differentiated tumors and knocking down either a control construct, in this case luciferase, or various basal genes such as p63 or p-cadherin. You can see that our knockdown protocol did not prevent invasion. Luciferase knockdown vigorously invades and vigorously activates keratin 14. But depletion of this single intermediate filament protein was sufficient to block collective invasion in culture and in vivo. So we think we've demonstrated that cancer invasion and metastasis require changes both in the molecular programs of the cancer cell and in the microenvironment. And the last little story I want to tell you here is focused on this connection between collective invasion and distant metastasis. Because I told you at the very beginning, metastasis is the key problem. And then the 2 sections on tumors, we're talking about the invasive process, which is an event that occurs in the primary organ. And so here's the thing: is how does collective invasion relate to distant metastasis? The still dominant EMT model proposes that single cells leave the primary tumor, travel as individuals, and found clonal metastases. And so what is collective invasion? Does it also give rise to single cells that found clonal metastases? Because if so, it may not really, in a deep sense, be the important feature. It may really only be this single cell. However, we had a couple of clues, both from our own lab and from the work of others, that this might not be the case, that there actually might be something specific to the collective invasion process that generated a different program for metastatic spread. And the first was that keratin 14 was required not just for local invasion, but also for distant metastasis. If we knock down keratin 14, we essentially abrogate the ability of these cells to metastasize to the lungs. There was also a few different data points over a long period of time suggesting that there was a multicellular intermediate that could be founding metastases, that there was a multicellular metastatic seed. And the first is that for as long as people have looked at the blood of cancer patients with compound microscopes, they've seen clusters of tumor cells. Lance Leota showed in 1976 that clusters of 6 to 10 cancer cells are much more efficient experimental metastatic seeds in experimental models. And there's a couple of papers recently, parallel efforts that published first, where they showed that multiclonal seeding is possible and likely predominant in experimental cancer models. There's also work showing that patient metastases show genetic evidence of polyclonal seeding between the different metastases. So collective invasion, instead of generating the same single cell intermediate, could in fact generate a multicellular intermediate that then traveled to the distant site. So we decided to distinguish these possibilities of single cell versus multicellular metastatic seed using a mosaic heritable fluorescent labeling strategy. So we have all the cells in red, Cre recombinase turns some of them green. We transplant that unit to found new mammary tumors. And the logic here is that if it's always a single cell traveling, we should only see single color single cells and single color focal proliferations of those single cells. Whereas if they invade and disseminate collectively, we should be able to observe multicellular, multicolored intermediates along the process. So let's start. If we look at the primary tumor, it's multicolored, multiclonal. The collective invasion strand, again, multicolored, multiclonal. We can see locally disseminated nests in the primary tumor stroma, multicolored, multiclonal, multicolored circulating tumor cell clusters isolated from the blood. And then when we look at the distant metastases at the micro and macro metastatic scale, what we again see is multicolored, multiclonal metastases. Our computational model revealed that more than 90% of these cells, 90% of these metastases, from more than 1,000 examined, were likely founded by multicellular intermediate. So the final question we really wanted to address was the relationship between progression and keratin-14 status. When was keratin-14 expressed during each of the steps of metastatic spread? So we were able to show that it was rare during the growth of the primary tumor in this model, ubiquitous during collective invasion, local dissemination, circulation through the blood, and the micrometastatic stage, and rare again during outgrowth of the metastatic tumor. We're actually able to model this keratin-14 seed forming a keratin-14 negative metastatic outgrowth in vivo as well as in culture. So here's 3 keratin-14 positive cells that are founding a mixed colony with a keratin-14 negative proliferative core. So we propose from all of these experiments a collective epithelial path to metastasis, where groups of cells in the primary tumor acquire this basal epithelial phenotype, collectively invade, collectively disseminate. They're maintaining epithelial gene expression such as E-cadherin, requiring epithelial gene expression such as cytokeratin-14, and then re-expanding to a proliferative keratin-14 negative phenotype at the distant site. And I think it's important to say that this need not be the only path to metastasis, and it's going to be very important in the next few years to understand the relative frequency of these collective paths to metastasis versus the single cell EMT path to metastasis. I want to close here by thanking the wonderful students who've done this work in my laboratory, my postdoctoral fellows, both present and former. Ryan and Kevin both have their own labs now. We had great undergraduates and terrific collaborators across the clinical disciplines and the engineering disciplines. And I would love to hear any questions you might have.
Yeah, sure. Thank you very much, Dr. Ewald. So it's a very nice presentation.
I try and figure out how to unshare my screen.
Okay. Yeah.
Can you see me again?
Yeah, I can see you now. That's good.
Perfect. Thanks.
It's a very nice presentation. I have a couple of questions.
Great.
How do you correlate between, like, you had initially mentioned about matrigel and what is the other collagen one, right? So what is the relationship between these cadherin and other molecule KL14 with those? Is there a relationship between those two in disseminating and migrating?
Right, because the simplest idea here, I'll say it without the slides, but we have some nice slides on that. That's present in the cell paper. The simplest way of interpreting that is to say they only invade into collagen 1. They don't invade into matrigel. So therefore, your starting hypothesis, which I bet's what you're thinking, is that maybe they only activate keratin-14 in collagen 1. That was what we thought. It's not, in fact, correct. They'll activate keratin-14 at the edge of the tumor in matrigel, in collagen 1, in collagen 4, in laminin, as well as in agarose. The agarose results suggest to us that they're not detecting something specific about the molecular composition of the tumor border, but instead what they're detecting is the absence of cell-cell contacts. So what we're actively pursuing now is the molecular basis. What's the sensor that's telling them, I don't have a neighbor there, maybe turn on this program. And so we're working hard on that now. But they'll turn on the program across different matrix environments. They'll only invade into specifically fibrillar collagen 1.
And related to that, I mean, like it may be putting in a different way. Like, for example, in breast cancer, HER positive and HER negative, HER2. So how does this positivity of HER connected with some of these molecules? Is there any, like, HER increase will indirectly increase the KI14 or something like that?
This is what we're actively working on with pathologists, both at Yale and at Johns Hopkins. But what I can say now is that we have observed keratin 14-led collective invasion across luminal A, luminal B, HER2 positive, and triple negative breast cancer. So this is something that appears to occur across the molecular subtypes. Our interpretation of this is that the molecular subtypes as currently defined are very accurately defining the control of proliferation in the tumor, and they're accurately helping physicians understand which proliferation-targeted drugs to use, right? Do you use Herceptin? Do you use endocrine therapies? But it appears that there's an orthogonal control of metastasis, which is not neatly falling into those bins, and we need to understand. It's very important, for example, in the future to understand, is there 1 or 2 or 5 molecular programs for metastasis? Are those evenly represented across the different molecular subtypes, and how do these proliferation and migration metastatic programs relate to the founding mutations that are occurring in the cancer cell? And it's very early days for that. We don't have complete answers.
Okay. And finally, like, this is a kind of a basic, may not be related to the the molecular aspects. This is kind of a breast cancer, which you see every other family, somebody or the other is related or like had some issues with the breast cancer and all those things. So like one of the things when I talk to the doctors, they say, okay, it originates at one place. It might be, there may be many origins for these breast cancers. So if you eliminate them early on, you may not be able to see them later on in your life. So is there anything like kind of like multi-origin or like originate, okay, there is at one place in the tube, there is only some cells at one region might be starting to grow, not at different, like if you eliminate it, you find out that, okay, this is the region which is starting to proliferate and you identify through various technologies currently available. You may not get it in other places or people don't know about it, still it's too early.
So there are some statistical differences in terms of the different regions of the mammary epithelium that are more or less likely statistically to give rise to tumors. But it's not a strong enough correlation that you could really use that clinically. There's also very many ducts. There's multiple different ductal systems per breast. And so there's very many sites of where a tumor could potentially form. Current screening methods work, right? They do reduce mortality. If you look at the past 20 years, breast cancer mortality is going down. They don't work as well as we would like. One of the really, I think, terrible things about breast cancer is that some of these tumors come up very, very quickly. So even people who've done everything right, they've exercised, they've seen a doctor regularly, they have had their mammograms regularly, they can have a tumor come up in between their mammograms. They just came up very quickly, spread very quickly, and can be very lethal. Likewise with the metastases, either early, as in the triple negative, or late with ER positive, it can be years or decades later, nothing, 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 and then like wildfire, it spreads. So what I can say is that we're working very hard on this. We're working collaboratively with physicians across different disciplines, surgery, pathology, medical oncology, and radiation oncology. We're working to understand the clinical problem and as aggressively as possible work to fix it. We're working at the same time in collaboration with physicists and different types of engineers to build the absolute best model systems and then we rapidly communicate those results and collaborate nationally and internationally to really work on this as a team. It's not ultimately very important who gets credit for which step. What's ultimately very important is that we make progress, work as hard as we can, work as together as we can to make progress against this terrible disease on a time scale that can be relevant to patient outcomes for patients who are alive today. So that's our commitment.
And finally, like when we were talking, like something came up in my mind that like, are these some of these molecules, like you are talking about dissemination, migration, and all those things? Are some of your like KL14 or in some of those ecadherine and all, can they be used as markers early on to figure out, hey, is there a possibility of these eventually finding their way out? Like as a marker, you might get into eventual very strong metastasis or not.
We're working very actively on testing the utility of keratin-14 as a prognostic biomarker for future metastasis. There's a study from Philip Rudland suggesting that it might be very suitable. The challenge that we're thinking through is that you might only need a few cells to be positive in order for the tumor to be sufficiently metastatic. So our current data strongly suggests that the biology of keratin-14 positive metastatic spread is very widespread across different types of breast cancer. What we're working hard with some of the best pathologists in the world on is how to decide what would be the threshold. Where in the tumor would it be more or less dangerous? What fraction need to be keratin-14 positive? These sorts of more refined biomarker decisions and strategies that I think will be necessary for it to be clinically useful. And that's where I think of translational research as a relay race, right? If you watch the summer relays in a few weeks, you're going to see the best relay races. Man, they hand that baton to the next person very efficiently, right? They never drop the baton. They never hesitate. They get the guy right in stride. And so that's how I view this. I'm not a pathologist, and I never will be. And so when we have a basic science laboratory observation that might be clinically important, we just immediately go and try to find the best minds in the world on that. And we've been very fortunate that people have been very willing to work with us. So to be continued. It's not perfectly easy to do, but we are actively working on it.
Okay. So thank you so much again for your expertise and bringing out your research to the scientific community in through Science Hangout. I really appreciate you coming on to Science Hangout, and hope with your new results in coming months, you'll come back again and present your new results. Thank you very much.
You're welcome. And I want to say thank you to people in the audience too, because my laboratory has been funded both by federal funds and also by combination of foundation and philanthropic funding. And so it's something where everyone who looks around and sees those pink ribbons, the American Cancer Society, the Breast Cancer Research Foundation, the Susan G. Komen Foundation, they're funding our lab, they're funding this work. So people who have walked a Relay for Life, who've done a walk or a race or a run for the cure, they're really directly contributing to the research in my lab and enabling the research I just talked to you about. So I want to say thank you to them for making all this possible.
So thanks again, and I'd love to see you more of your work in the journals, reputed journals soon.
Great.
Thank you.
Thank you.